Terrific. So the title is Where John Spence's Vision Led My Research, Demonstrating New Techniques for Watching Biomolecules at Work. And I should say that John really has been an important force in my work over the last decade plus when I first met him, and you'll hear a little bit about that in here. So it's a real honor to be here to kind of honor his contributions, at least those that have benefited my research. So. Okay, so now, all right, I think I probably have to click here. Now we'll try it. Great. So here's the outline. What I'm going to do, since this is a talk about biophysics, I'm going to start by introducing you a little bit to my favorite biological molecule, which is RNA. And I know this is a physics audience. I will give you all the background you need. Please don't worry about it. And I'll motivate the need for new tools to measure RNA structure. And then I'll talk about the technology developments we've made, mostly using X-rays to further our understanding of how RNA works. And I'll tell you where we are and what's next. So let's start just with an introduction to the three major molecular players, the molecules of life, if you will. They are DNA, RNA, and proteins. And these are very simplified representations of their structures. And just to give you an idea of kind of what they look like, maybe you can see some similarities, maybe some differences, very coarse representations, but there they are. DNA on the left, RNA in the middle, proteins on the right. But let's talk about what they do a little bit, the functions of these molecules, right? So DNA, as you may know, is kind of the genetic blueprints. It's like I view it as an encyclopedia that contains the information for manufacturing all the molecules that your body's going to manufacture that help support life. And for a very long time, people thought that those were just proteins. Right? So again, following this DNA on the left and proteins on the right. So proteins were thought to be the molecules that carried out all of the biological functions, helping you digest your lunch, structural pieces, I mean, just everything that was needed to support life. And in this early view, the third one, which was RNA, was viewed as a messenger because the DNA is so important and contains such critical information, it has to be kind of sequestered away. I view it kind of as a reference library, so it's in the reference section, it can't come out. And so you need to have something that comes in, copies the information from the DNA, so the DNA gets copied into RNA, and then that information comes out of the reference library into the cytoplasm of the cell, where it can be translated into proteins, which were thought to carry out all of the biological functions. And this is kind of the central dogma. Everybody thought, yeah, this is the way it works, and RNA in the middle is relegated to being this messenger. But with time, it became known that only about 2% of what was written in this encyclopedia actually made it all the way to proteins, right? And so only 2% of that information coded for proteins. And so for a while, everybody thought, well, the rest is just junk. It doesn't do very much. But surprisingly, 80% of it actually got turned into RNA, right? So a lot of this DNA is being made into RNA, and that's launched a whole field, right, trying to answer this question. There's a lot of RNA in cells. What is it doing? And the answer is a whole lot. Okay, so I've told you in very brief terms about structure, and then I've told you about function, the jobs that these molecules have. And there's a very important connection in molecular biology, the structure-function connection, which basically goes like this. If you know what the molecule looks like or how it moves, there's very important clues about what it does. So the structure or the shape of the molecule, by looking at it, you can figure out what it does. And here's kind of a more familiar example, slightly irreverent, but there we go. If you see something like that, right, you know what's coming next. It's really clear what that structure does, okay? So that's just so that's kind of what we're after with these molecules. We want to see what they look like, and then we can deduce or guess at what they do. All right. So if we now delve a little bit deeper, picking up these pictures from my first slide, we can see that, in fact, DNA RNA resembles DNA structurally. So we have these helical forms over here, and there are things that look very much like them in RNA. But we also see that it's not as rigid. So these helical pieces over here, these so-called duplexes, are very, very stiff and rigid, right? They have a long persistence length. They don't bend. But these other parts of the molecule are very flexible over here. And so because RNA isn't as rigid as DNA, it can fold into more compact structures. And here it kind of resembles what we associate more with proteins, where there's rigid parts over here, helices, and there's loops. So the bottom line is, from looking at these structures and what we know about both DNA and proteins, we can make some guesses. Actually, they're not just guesses. It's known that this is what RNA's functions are. So because RNA has some of the same properties structurally as DNA, RNA can store and transmit information. That's DNA's role. It's an encyclopedia. And RNA kind of looks like that. It makes a copy of what's in the encyclopedia. So it's not a surprise that, like DNA, it can store and transmit information. Although I add parenthetically, not as securely as DNA, right? DNA kind of has to live for a long time. It's the instructions. RNAs have a much, much shorter lifetime. Okay, and it's fine that that's what they, that's perfectly aligned. They take the information, they bring it out, proteins get made, and then the RNA gets degraded, part of the job. All right, but in fact, because RNA can also fold like proteins, I showed you that in the last slide, it was actually a huge surprise to the field, garnering the 1989 Nobel Prize to Tom Cech and Sidney Altman, that RNA could actually perform jobs like proteins could, right? It was thought that just proteins are the only ones that can perform functions or catalyze reactions in more biospeak. But because RNAs can fold up like proteins, it has a whole bunch of jobs to do, though I will add not as elegantly as proteins, because RNA has a more limited set of building blocks. So in fact, RNA has all of these roles out there, and there's many, many more roles yet to be discovered. And I say discovered because quite often, it's been about every 10 years, there's this remarkable new function that people discover, such as RNA interference, but I don't think I have time to go into that. And it's just something that we never even thought of, and there it is. It's discovered. So RNA is incredibly versatile. All right, so now I have to tell you a little bit about RNA structure, right? Just before we get into, kind of the details of the talk. So RNA is a polymer, right? And a polymer just means that it's a chain molecule. It's built up by adding different monomers, like beads on a string. And so it's constructed from 4 distinct but chemically similar building blocks that are called nucleotides. And each nucleotide, each monomer or building block that gets strung together in some sequence into a chain of an RNA molecule, it consists of a piece here, a phosphate sugar group, and a base that gets stuck onto the side. So that is the fundamental building block of RNA. And there's 4 different types of bases, G, Cs, As, and Us, which you may know from the DNA analogs, G, C, As, and Ts. In RNA, it's a U, not a T. But these monomers get strung together onto a strand, and that's what makes up the fundamental RNA strand. Importantly, each one of these phosphate groups carries a negative electron, a negative charge minus E. So RNA is wildly negatively charged, which makes it kind of a playground for physicists, really, because it's largely an electrostatics problem, which is really remarkable and fun. Okay. So like DNA nucleotides, I mean, I hope people know about base pairs. G goes to C, A goes to T in DNA, right? That's very, very fundamental. RNA nucleotides also can pair together. So we get GC base pairs, we get UA base pairs over here, and the bases can pair, and then they can stack. And this is responsible for this beautiful helical form that I've been showing you, double-stranded helix or a duplex, which is what I will call it. And these are very rigid structural elements, right? They're like sticks when you look at the structures. Okay. But like proteins, because it's flexible, RNA can fold up. So if you took a DNA duplex and you grabbed it and pulled it apart, you would end up holding two separate strands. But in RNA, because they're copied from DNA, they're primarily one long strand. So RNA is a single-stranded molecule. Over here, this is kind of a copy, a transcript, a copy of DNA with the nucleotides arranged in a certain order and a negatively charged backbone. And what happens here is that one piece of this will have a complementary strand on another part of the RNA. And so this single-stranded molecule kind of folds up into these helical or duplex regions where there's complementary sequence, and then there's flexible parts here that aren't complementary. And when you add magnesium ions, very, very special relationship between RNA and magnesium, you can get this molecule over here to fold into a beautiful three-dimensional structure. And quite often, these folded structures are catalytically active. So this molecule might have a particular function. RNA likes to cut things up. And so this might actually look for a particular other molecule and then chop it into pieces. And that can be a very important function. So RNAs come in many, many sizes and shapes, but I am particularly interested in small RNAs. That's RNAs that are less than 60 nucleotides long. So these chains with smaller than about 60 monomers on them. And that's because RNAs, they like the same kind of motifs. So if you find something interesting in a small RNA, chances are, if you look at a larger structure, for example, the ribosome, which is a huge RNA machine, an RNA protein machine, you can find repeated motifs or patterns. You can find an interaction in the small RNA that's reused in these bigger molecule. So if you understand the smaller molecule, you can kind of get the playbook. This is how RNAs like to interact with other bits of RNA. They're easy to get. You can make them or you can buy them. And they're amenable to atomically detailed simulations. So I work with some groups who do atomically detailed molecular dynamics simulations, and this helps us interpret the experimental data that we take. It's actually quite important. And again, these small RNAs are highly dynamic. And quite often, I told you that they don't have to be subtle. They can operate by not very subtle conformational changes. And here's an example. So here's a kind of a schematic of how a small RNA motif works over here. This is a sensing element. So again, here, this region that looks like a ladder is just another way of showing this duplex, right? I know there's lots of different ways of showing that, but the job of this molecule over here is to bind this small ligand into a pocket. So when the ligand binds in the pocket, that actually triggers a whole reorientation of the molecule. It kind of refolds the molecule. So instead of having this duplex down here, we get a much longer duplex over here, which in fact acts like a molecular crowbar. And once that big duplex forms, it crowbars another molecule off and stops a process, right? So that's what I mean by not always very subtle. And so I am very interested in how RNAs sense these ligands. So what is it about this ligand that allows it to be specifically recognized by this molecule? And then how does that binding trigger this, kind of rearrangement of the molecule? And this is a pretty large scale rearrangement, right? We're moving this duplex into this much larger one, which then affects, kind of a very significant change in biologically. Okay, so again, I'm interested in how the molecule goes from this initial state over here, from this initial state to the final state, right? And questions about on the small scale and then on the larger scale and kind of the dynamics of how this happens. All right, so because people thought, and rightfully so, that proteins are very, very important molecules, there's a lot that's known about proteins and protein structures. And there have been phenomenal methods developed for measuring protein structures. And those include crystallography, NMR, cryo-EM, many, many wonderful tools that you have access to here at ASU. And because of that, there's more than 200,000 structures that are deposited in a repository, kind of shared data. The structure of more than 200,000 proteins are known, mostly with atomic detail, and that's pretty amazing. And because of all of this knowledge, atomically detailed simulations work really well for structured proteins. And there's even new approaches. Maybe some of you have heard of AlphaFold, right, which is kind of an AI machine learning, trained approach. And this is really great. So, structured proteins are, pretty well understood, although one can always be surprised. But much less is known about RNA structure. So there's only about 1% of the amount of structures, about 200, it should be 2,000 RNA structures in the PDB. There's a typo here, 1% of proteins, so a small number. And that's because RNAs are highly charged and intrinsically dynamic. And it makes them hard to study by the methods that work for proteins. A lot of times you need to immobilize the proteins, and I'll show you a little bit about that later. And because RNAs are so highly charged and so mobile, it's hard to immobilize them. And there's some references if you're interested in more details about that. So we need new approaches to determine the structures. Okay. Now we're switching to the next part of the talk, which is the experimental part. So X-ray scattering is my group's our method of choice, and we use it in two different ways. So macromolecular crystallography, you can make a crystal of these biological molecules, just like I have a crystal of carbon known as a diamond on my hand. You can actually get these molecules to arrange in a fixed orientation, and you can grow a crystal of proteins or RNA. And when you shine X-rays on this, because the molecules are all oriented, you get very, very high resolution, kind of angstrom scale, which is atomic scale. You can measure a structure with atomic resolution this way. But the problem is that by forcing them into this crystal, you're limiting their flexibility. So you sacrifice a little bit on the motions to get the resolution. On the other hand, you can do solution scattering, where you have the molecules freely drifting around in some kind of buffer. And here you have high structural flexibility because you don't have to worry about the molecules being, they're not locked into an orientation. But because of the spherical averaging, you reduce your resolution, okay? And I actually want the best of both worlds. So I'm interested in the sensitivity to these large dynamics. Remember, I showed you that some of these rearrangements involve, fairly significant structural changes in the molecule. But I'm actually interested in high resolution as well. And that's kind of a hard place to go. But anyway, I'm going to limit most of the rest of the talk to solution scattering, because that's what we're trying to do. All right. So solution scattering, it's a really powerful technique to apply to small, highly dynamic molecules. Traditional solution scattering is called small angle X-ray scattering, because you have X-rays coming in and you scatter them to small angles and a couple degrees, something like that. And the resolution of a small angle X-ray scattering experiment is typically hundreds to tens of angstroms, not the atomic, the single angstrom resolution that we're after. The molecules, however, are free in solution. You don't have to constrain them. You don't have to label them. That's a plus. The measurements can be at room temperature or any other temperature. It doesn't really matter. The dynamics are easily measured because these molecules flop around. They're free and they're flexible. And you can trigger reactions by either changing the chemistry or flashing with light. And finally, my favorite feature, I think, you see the full ensemble. So these molecules do not exist in one rigid form. They're actually moving. They can, they have a certain variation in their structures, and that's actually quite important. All right. So this is the experimental setup. So we have X-rays coming in from whatever source we want. And there's the sample. So the X-rays come in straight. They interact with your sample, which scatters them. They come off at all kinds of different angles over here, and we measure the scattering of the X-rays from the sample. So you have something that comes in straight, gets scattered, and the game is, how do you figure out what was in this box to change this straight beam into this scattered beam, right? That's the whole, kind of thing about X-ray scattering. And then what we do is we actually take the scattering profile. This is an image over here, and we go to a particular scattering angle here, 2 theta, and we just count all of the X-rays that are scattered to that angle. And then we put a point over here. We go to another angle and do the same thing. And we plot the scattering angle as a function of Q, which is just, we plot the intensity as a function of angle, essentially. And I will say that the units of Q are inverse angstroms, just to point out that when we're at low Q over here, we get, so small in inverse angstroms means big in angstroms. We get information about the largest size, the size of our molecule here. And when we go to high Q over here, right, large inverse angstroms means small in real space. That's where we get the high resolution when we go to high Q. Okay. So from small angle X-ray scattering out to 1/2 degree or something like that, from the scattering at the very lowest angles, you can get something that's related to the size of the object. That's the largest spatial dimension. And just to give you a sense of why that's important, when we're looking at reactions with RNA, the size can change by almost a factor of 2. So it's a good thing. You actually have sensitivity here to the kind of reactions you're interested in. But the full profiles contain a lot more information. And in fact, if you look at the intensity versus Q for 2 different states of the molecule, my eye, and maybe Rick's eye, and some other people in the audience here, might notice that these curves are really different. So, and I'll forgive those of you who don't see those differences, so I'll help out by plotting intensity times Q squared versus Q, basically multiplying this by a parabola to enhance the signal at the higher Q. And now you can all see that the folded molecule looks very different from this unfolded molecule, right? You get a big peak here. And so overall, from small angle X-ray scattering, you get information about the size and shape through the size at the lowest angle and the compactness. A folded molecule is compact, an unfolded molecule is not compact. So that's great. So where do our X-rays come from? So we have a home source, and sorry, there's about 5 of them in the complex that I work in. It's called the PSB complex. So these are really common things, right? We also can get X-rays from synchrotrons, and these are national facilities. And there's about 5 in the United States, including one that we have at Cornell called CHESS. And our X-rays can also come from X-ray free electron laser sources. There's about 5 in the world. And soon, you here at ASU will have another type, thanks to Bill and his group, the compact X-FAL will be just in a nearby building. So at some point, maybe I'll use that, and we'll add that to my list over here. Okay, so the X-rays from each source have distinct properties. So just to give you a sense of the comparison of the sources that I use, from our lab source, we get 10 to the 8th photons per second. This is not scale. It's about 1 millimeter across the beam that I get from my lab source. At a synchrotron, we get 10 to the 12th photons per second into something that's about 100 microns, 1/10 of a millimeter by 1/10 of a millimeter. At the X-FAL, we get 10 to the 14th photons per second into that teeny tiny dot, okay? So the number goes way up and the size goes way down, and that's going to be important. All right. The other bit in this experimental section that I really need to mention is that the background subtraction is critically important for doing solution scattering, because I told you that when the X-rays hit that sample, they scatter. and you want to find out what it was that you put in the beam to make them scatter that way, you didn't just put your biomolecules. There's actually a, some kind of a holder, and there's buffer, and there's all kinds of other things. So to figure out the scattering from the sample alone, these little biological molecules, you have to be able to measure the scattering of everything, and then you have to subtract off the scattering from everything that's not your biomolecule, and you get your sample alone, okay? And so that's the background subtraction. All right, so that's how we get spatial resolution, but how do we get time resolution? Remember, I told you that I want to watch these molecules move and fold up and change shape. So we do that with microfluidics. And so for actually quite a long time here, when I was a postdoc, I built the first one of these devices for X-rays. These are microfluidic mixers, and they're really kind of cool. So in the simplest case, we take two cross channels and we etch them through a silicon wafer. And then we put windows on, and then we drill holes on the end. And then we can flow fluids into each of these 4 ports. So we flow fluids into 1, 2, 3 ports, and it flows out through that exit port over here. So we have a large molecule in one solution over here that we flow into the inlet port. We have a different solution, solution B, that we push from the north and south ports, and we hydrodynamically focus this macromolecule containing solution into a very thin jet that just keeps streaming out to the right over here. This is a fluid flow device. And now we let physics help us, because these molecules, these macromolecules, are big, so they don't diffuse very far. But if solution B contains small ions or ligands, right, and they are not present in solution A, anything very small can rapidly diffuse across this jet, and that's how we trigger our reaction. So our RNA, our protein, whatever it is, we can put anything we want in here, including crystals, and I'll get there. They flow through this very thin kind of micron-wide jet over here, and small components, small chemicals that are present in B and absent from A diffuse across the solution, triggering our reaction. So adding the magnesium to fold the RNA. That's why I told you we have to have magnesium fold the RNA. And then we come in with whatever beam we want, and we sit and we look at any location here. And that is a measurement of fixed time after the mixing happened. Then we go to another place and another place. Or we can sit at one place and then change the pressures or flow speeds. That's another way to change the time. Okay, so we've used mixers of this type at synchrotrons, such as CHESS, to probe RNA folding. And because I told you that stream is about a micron wide, right? That's a pretty small sample. And if we want good time resolution, we want a very thin beam, right? Because we're measuring across the jet. So we just want to take a small sliver of it, the proportionality factors of flow speed. So you really need a small sample and a small beam to get good time resolution, okay? And so because of that, we're limited in our spatial resolution to a relatively low scattering angle here. And that's just because there's not a lot of sample and these signals fall off quickly. But nevertheless, we've learned that the relevant time scale for these reactions are milliseconds to seconds. And in fact, we can use molecular dynamics to get a ton of information from this. I'm not going to talk about it here, but this has been a really fun and successful approach, but it relies on the MD simulations to kind of help us interpret this data. All right. So what I really want to do is see if there's a way to increase the spatial resolution of these time-resolved measurements so that we don't have to rely on the MD. And also, yeah, I'll just go on to this one. So Sachs profiles are wonderful. I love them, but they're largely featureless. So this is just a monotonically decaying curve, right? And so you can look at that and think, well, I can fit a lot of things through there. It's not ideal. You get a lot of information, but it's not ideal. But if you go out to wider angle here, all of a sudden you start seeing these bumps and wiggles that appear, okay? And so this I like because these are features and they give us information more than just a monotonically decaying curve. All right. So at this point, we measured that, and I said to my students, "What did you do?" Right there's no bumps there. Go back, remeasure that. But it was very clear that those bumps and wiggles were coming from the sample. And so I went and looked in the literature and found that, not surprisingly, people had been doing this wide angle scattering for a while and pushing out to higher scattering angle here. So it turns out that a Q of 3, something like 45 degrees. So these are huge scattering angles out here. And up here, just to give you a sense of a feel for resolution, this is the crystallographic conversion of this Q into space, which isn't exactly correct for solution scattering, but it gives you a sense that we now have a resolution of about 2 angstroms here, right? 2 angstroms. That's pretty good, right? Atomic resolution is about an angstrom. So we can do this. And this has been applied to proteins and also to DNA. And it was this wonderful paper from Dave Teedy's group at Argonne that really caught my attention with a particular kind of a DNA duplex. And in addition to this large monotonic decrease, all of a sudden these bumps and wiggles appeared. Okay. So what are the bumps and wiggles? Here I went back to the basics, and this is one of my favorite books, X-ray Diffraction by Warren. I have a really old copy in my office, and here it says, peaks and dips in a scattering curve do not require crystals. The mere existence of certain interatomic distances, which are more probable than others, is enough to add these peaks and dips. And so if you go back and look at the structures of DNA and RNA, oh my gosh, there's repeating distances and length scales all over the place here. And that is what is reflected in those peaks in the wide angle scattering profile. So that's what we measure. So how do we interpret these peaks? Well, one of my former graduate students really got excited by machine learning and AI. And so he trained a machine learning model by comparing molecular dynamics generated structures and computing their small and wide angle scattering profile and matching up the peaks and valleys with the things that he could see in the structures, and then fed them all into a machine learning algorithm, and came up with something. And he walked into my office one day and said, "That's the helical radius, that's the major groove," and could just tell me what each of those features were. And that was pretty cool. So this kind of occurred to me to be a promising approach. We could use small angle X-ray scattering to create an outline, and then we could use wide angle scattering to fill it in. And so, again, we're always going to work with MD groups to improve it, but we really wanted to explore the sensitivity of wide angle scattering to RNA structures, right? Nobody had really done that before. So we went to another synchrotron, NSLS. And so we started with, I don't know, I didn't really know where to start. So we took the simplest RNAs that I could think of, which are single-stranded RNAs. They're homopolymers. Here, this is poly-RU, which is 30 strands of U's all in a row here. And then we made another one, which is 30 strands of A's all in a row here. And then we did a lot of work on these, and we knew from other work that we'd done with other techniques that in fact they had different structures. RU30 is pretty much a random coil structure, but RA30 likes to make these little curly Q helices over there. And when we measure the profiles out to now a Q of 2, you can see there's the part that just comes down from the sacs. And here in the poly-RU, well, there's something, and then the RA, maybe there's a couple bumps, but this really, it's not that convincing, right? I mean, it's nice, but I want, better resolved features. And so we tried more structured samples over here to get these length scales in there. And so we wanted to take a duplex. I've told you a lot about duplexes, but we can actually make them even more rigid by adding a third strand that kind of threads through the groove. Okay, and that's called a triplex. We'll get there in a minute, but But here's a base pair over here, right? A U and A base pair. Turns out that in many cases, another base can come in and kind of bind to the backside over here. It binds to the outer edge. And this is many of the ways that these ligands, remember I showed you this ligand early on that kind of comes in and lands? A lot of them have these base triple interactions. So this is getting more biologically interesting. So these triples are found in many biological RNAs. And when a number of them stack up on top of each other, that's called a triplex, right? Which is a 3-stranded nucleic acid molecule. Okay. So my student Yen designed a duplex over here, which could be converted to a triplex by just adding another molecule. But already here, we started to see some more significant bumps. And through the machine learning algorithm, Yen was able to tell me that that bump over here corresponds to this particular length scale in the molecule, the major groove. But when we actually put that third strand in, all of a sudden, we got this whole forest of lines out there. So now, we're starting to get things that we can measure. And so we know from simulations and from some work, we haven't done the ML out here yet, these peaks correspond to lengths along the backbone. And this little tiny peak out here at about a Q of about 2, which is about 2.9 angstroms out here, that corresponds to the base stacking distance in these structures. All right, so I need to remind you that these signals are measurable, but they're weak. So here's the SAC signal, right, from that molecule. And you can see, right, there's that part that just comes down and it's very smooth and monotonic. And way out here, right, this is where the triplex features are. It's this Q out here. And you notice that we're about 100 to 1,000 times weaker. So these signals are there, but they're not very strong. Okay, so wide angle scattering is a challenge for static experiments, right? And remember, I really want to do time resolved. I want to start with one structure and see it evolve to another one, right? Because this is how we can learn about the physics of these interactions, maybe even capture reaction intermediates. And I don't know, was this even possible to do? Okay. So, all right, I wanted to design a system that would start from a single strand. Remember, these single-stranded molecules don't really have much structure. There's kind of a, a loose peak out there. And then we add magnesium and it folds into something really structured. So here we have a system that starts with a loose peak, but ends up with these really fine peak, 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 baby peak. That's what we call them, right? And so this was our test system. Okay. So now signal strength was going to be the problem, right? So as I told you, these wide angle signals are 100 to 1,000 times smaller than SACs. And now if we're going to use a mixer, these jets are microns wide. Okay, so we had a huge sample cell out at Brookhaven at NSLS when we did it. So the only way to do this is with more X-rays. We can't use synchrotrons, at least not the present generation of synchrotrons for this. So this is where we turn to the XFEL, right? And so many of you may know quite a bit about the XFEL. It's the one that we use is out at Stanford, and it's a really beautiful machine. There's a long accelerator, and then the electrons get wiggled and X-rays get created, and they come all the way down here, and our experiments are carried out here, a couple of miles away from where they, began way up here as electrons at the CXI beamline. Okay, and so the X-rays from an XFEL source are different from the X-rays from a synchrotron or from our lab source. They arrive in pulses, and they're tens of femtoseconds long. So, I used to play tennis, and there were these machines that would kind of have balls coming, and then I would practice my forehand or my backhand. That's what it reminds me of. These balls get kind of launched at you, but they're short. 120 of them come per second, and each one carries 10 to the 12th photons, which is as many as I get in 1 second of beam at chess, right? And you get one diffraction pattern per pulse. And for me, I don't take advantage of the timing. So the femtosecond part, I don't really need that. I just need the flux. I need as many X-rays as possible in a little tiny package, right? So other people do experiments with the timing, not me. I just want the flux. But there's challenges. And I saw this outside John Spence's office upstairs, destruction, which means that when you hit something with that pulse, you blow it up. But as we heard from Dave, the molecules diffract before they're destroyed. So it all works out. You get the information before the molecule blows up. And this is really, this is a beautiful first demonstration. The X-FEL pulses come down here. There's a liquid jet. A lot of the development work for that jet was done here at ASU. And here are crystals. This work was done on single crystals, like a nano garden hose that shoot these, this jet that shoots these individual crystals in where they get hit by a pulse, they diffract, and then they blow up. And this, of course, launched an entire field of serial femtosecond crystallography, which with much of the development done through ASU. And here what happens is you get one pulse, one crystal, one diffraction pattern, boom. That's the way it goes. You blow it up. Okay. So John came to talk to me. John Spence came to talk to me in about, I don't know, 2011. And he came down to my lab and he said, this is a perfect setup for your flow cell. So we have this little, these little injectors, these gas dynamic virtual nozzles that shoot the crystals into the beam. Why don't we just put a flow cell in there, right? And see if we can do a mixing experiment. And that was really the start of my contribution to the BioXFEL STC, right? Here, that was largely headquartered out of ASU. And so all we had to do was figure out how to interface our mixers here with this existing gas dynamic virtual nozzle. And that's pretty much what we did. Okay. And so we built these. I've had a suite of really amazing graduate students through my lab. And they have designed these beautiful Cornell mixing injectors. That's what they're called. And here you can see that they're just concentric capillaries, right? Using a lot of the same technology that was used for those GDVNs that were built here. And you can see the colored solution represents where the sample would go. Hydrodynamic focusing, they mix, and then they get shot out where there's an intersection with the X-FEL beam, and then that's where you get your diffraction from. Okay. So with BioXFEL collaborators, we were able to demonstrate mix and inject serial crystallography with millisecond time resolution. So here we had protein crystals flowing in, right? And it was really great. And I know Alex Ross's group is working on similar approaches here using droplets here at ASU. And anyway, most recently, we've demonstrated this at a synchrotron. So there's the bioRxiv reference if anybody's interested. We did this at APS, which will make it more broadly available. But then I started thinking, could we use these injectors for time-resolved wide-angle scattering over here? So here's the injector over here. And so now what I want to do is put my little RNA in there, right? Flow it through. Mix it with magnesium, get the molecule to start flowing, and then zap it with the X-FEL pulse. In solution, no crystals. Okay, and then finally, we zap it, and then sometime later, it folds down here. So beginning state with kind of not so interesting wax signal, ending state, this triplex with these beautiful peaks, and we want to watch this as it happens. All right, so what are the challenges? Well, we need to do this background subtraction, get rid of the buffer and everything else that's going there. That's not so easy. Properly normalized profiles. Remember, each pulse provides one diffraction patterns. We need about 5 minutes of exposure. That's 36,000 frames. So we acquire 5 minutes of RNA and then 5 minutes without the RNA, just so we can subtract off the water. But the issues are that every pulse is different, and we've got 36,000 of them in there. The jet width, although it's quite stable, it's not perfectly stable, and things are always moving around. And we need this thing to be stable on the hour time scale because we have to load it, let it flow, and take data. So this is quite a challenge. And at the time that we started, the analysis software did not exist, right? But with the efforts of Tom Grant and Rick Kirian, it is being born, so to speak. Okay, so just a little bit about the data, because this is physics department, everybody wants to see data. This is what we get for a single shot, right? So that's one pulse going through, that's about what we see over there. And when we take 15,000 shots, this is what we see for the sample, and that's the background. So that's the sample, the red one. The background is blue. When we scale them and normalize them, that's the subtracted signal. But when we look in really closely, we get these beautiful peaks, right? So those are these wax peaks that tell us about the repeated distance. This is on a DNA control sample. This is my favorite sample because these peaks are just, we know where they are, we know what the relative size is, and it's just lovely. So this is the data on the RNA, actually. So this is real data now looking at the RNA. We start the initial state here, which has just kind of not very much structure in the wax region. 6 milliseconds after we add magnesium, we start to see this little feature down here, right, which is absent from the initial state and absent from the final state. So that is a signature of an intermediate. And then we start seeing these other peaks pop up here, right? There's peak, 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 and then way over here, we get the base stacking peak that comes up with time. And by comparison with models, we know we can interpret this is the radius of the helix. That's the major groove that tells us there's a duplex, backbone features, and the base stacks. So just by looking at these curves, without MD, just by looking at the curves, I can tell you that we start with a mostly unstructured molecule. The first thing that happens, because we capture this peak over here, is that 2 of the strands fold to a duplex. Then there's quite a bit of rearranging, and then finally it settles into the full triplex state here after about a second. Okay, so most recently, because I wasn't quite convinced about that little major groove peak, I made my student build me a mixer that got us faster to 2 milliseconds. Right, and there we go. We captured this in May. So this is a feature that is transient. We only see it on the most rapid time scales. So it's not present in the beginning state, not present in the end state, but it's there. So that's an intermediate. All right. So to summarize, we've now shown that this is possible at XFELS, time-resolved wide-angle scattering, and it provides new information, right? And so the first experiment was on a model system, but now, we're moving to real biological systems, and there are real important molecules that exploit these base triples for signaling. So, again, we've started working on, some very specific motifs, including these ribo switches, and that's the name of the class of molecules that I showed you at the beginning with the crowbar. So we're really excited about that. And so I will finish by acknowledging the people who contributed to it. And all of these XFEL experiments really take a whole village to do. But mostly the mixers here were built by my student Kara Zielinski, who was actually a summer student here in Brenda Hoag's lab many, many years ago. So she has a long history with BioXFEL. This is my current group, and it's just too much to read through all of these names. But by and large, a lot of the development was done through BioXFEL. And for my final slide, I just wanted to acknowledge the BioXFEL Center here, and it's really a center that's greater than the sum of its parts. And so here's a picture from our early days about a decade ago and about a year ago, and it's really been a joy to be on this journey. So thank you all so much for your attention.